Hello and welcome to our video on the synthesis of ethanol. Today we'll be covering a couple of things. So firstly, going over some of the common uses of ethanol both in society and normal everyday life. Secondly, ethanol can actually be made in two ways of significance to you. So firstly, through fermentation, which we're going to cover in the latter slides. But firstly, we're going to be covering the synthesis of ethanol through the hydration of ethene. So you may recall that ethanols are a type of alcohols. They have some common uses, ethanol specifically that is. We use it a lot in food colorings, perfumes, and especially antiseptics, so hand sanitizers for example. The ethanol or the alcohol in the hand sanitizer basically kills germs by disrupting the lipid or fat membrane. So if they have an incomplete membrane, the cell contents of that bacteria kind of spill out and it destroys the bacteria in that way. Another way in which we use ethanol is its use in E10 fuel. So you might have seen this if you're filling up yourself or your parents are filling up at the pump. That basically just means that fuel has a concentration of ethanol of 10%. So there's 10% ethanol in that fuel. So there are two main ways that we can make ethanol. So you can kind of remember them as one way being non-renewable and one way being renewable. So the non-renewable way of making ethanol is by the hydration of ethene. That's a reaction that we went over on our slides on reactions of alcohols, so feel free to flick back to that, but we do cover it in a bit today. Another more interesting way is the renewable way in which we can make ethanol. So ethanol is actually considered a renewable resource because we can actually make it by the fermentation of sugarcane, and this is a pretty cool reaction that we're going to be covering today. So before you continue any further, it's important to actually understand why ethanol is a renewable resource. And we need to have a conversation on what renewable and non-renewable resources are in the first place. So with a renewable resource, it's actually anything that can naturally replenish itself, right? So with ethanol, we can source it from the fermentation of sugarcane, right? And we can always plant more sugarcane and thus make more ethanol if we ferment more sugarcane, right? However, if we do that's the first method. If we do the second method here, so we hydrate ethene. Ethene is not a naturally replenishable substance. We can't quote unquote plant more trees to get more ethene. We need to chemically manufacture ethene. So thus, the second pathway is a non-renewable way of making ethanol. So here we have a summary slide on some of the advantages and disadvantages of the use of ethanol as a biofuel. So basically cars essentially running on 100% ethanol. And if ethanol is a renewable resource, that could be a possible solution to cars running on fossil fuels and petrol, right? So there are some advantages and some disadvantages for why we might or might not want to use ethanol as essentially a fuel. This was a big topic of focus in the last syllabus or the previous syllabus. I recommend you just glance over this and remember some of the key points to answer in bigger questions. However, I wouldn't spend too much time on it. Some of the key takeaways for the advantages being the fact that yes, it's a renewable resource. Also, we get less toxic air pollutants. So we get less carbon monoxide, soot in the air, less sulfur dioxide. Not only does that improve air quality, we also get decreased acid rain. And also, if we cultivate and distill it, and since it comes from renewable resources, it does end up being greenhouse neutral. Also, we need to look at it logistically, right? We're currently using a lot of fossil fuels. Those are non-renewable resources. Over time, the demand for those fuels will increase. The supply is going to dwindle and decrease. So thus the price of crude oil will skyrocket in the future. So we do need a kind of solution for this problem that's eventually going to happen. Whether you believe electric cars are the solution, that's one way. Biofuels such as ethanol could be another way, right? However, there are some disadvantages for potentially ethanol based fuels. So currently, cars can only take up a 10% ethanol burn, so hence why we use E10. And that is because ethanol, being an alcohol, tends to degrade the rubber tubing within the engines, and as well we need to kind of accommodate for ethanol-based engines. Currently, it's not economically viable. So petrol actually has more energy in it. So if you have octane, so octane is has a carbon chain of 8. So 1, 2, 3, 4, 5. 8. So that's essentially, that would be octane. I haven't drawn the hydrogens. That has more energy in it when you burn it than ethanol. And we actually spend, interestingly with ethanol, we spend more money cultivating it or fermenting it in the renewable manner than we actually get out of it as a useful fuel. So it's not that economically viable. Also, agriculture is a big thing. So we need to plant a lot of sugarcane, which inevitably is going to take up space. Space is already scarce with agricultural land, and if we're just dumping a lot of sugarcane, that poses a lot of problems. We did touch on this point of ethanol containing 30% of the energy that petrol contains, which means that needs to come up somewhere, because we need that energy to eventually propel cars. So what would happen is we need bigger sized tanks, and we need to make more ethanol, or you need to use more ethanol to travel the same distance compared to petrol, right? So that's a trade away for the fact that it's renewable. So one of the first ways we can make ethanol is via the hydration of ethene. So basically this is, as we said, the non-renewable way of making ethanol. This is an addition reaction where we're basically adding an OH throughout the double bond of the ethene, and we use dilute sulfuric acid or dilute phosphoric acid as the catalyst for this reaction. It's important to state some reaction conditions as well. So you need about 300 degrees Celsius and a pressure of about 60 to 70 atmospheres to maintain this reaction. However, this detail here is kind of loyal to remember. I would definitely just bother remembering the fact that we use dilute sulfuric acid and dilute phosphoric acid as the catalyst. So we did go over some of these reactions on our lecture series on organic materials, where we went over some of the reactions involving alkenes. So this is a very similar reaction to that. Essentially, the reason why we need a catalyst comes down to the reactivity of these alkenes. So alkenes will readily undergo an addition reaction with something like hydrochloric acid, for example. So as you may recall, hydrochloric acid would have a slightly negative charge on the chlorine, because chlorine is a lot more electronegative than the hydrogen. So thus the little electrons in this bond will spend more time with the chlorine than they will with the hydrogen. So you get that shift in the electron cloud. So that gives hydrogen a slightly positive charge. However, with water, that isn't entirely the case. So water kind of looks like this. So although there is some polarity, it's not strong enough for water to actually attack that double bond, if you like, by itself. So it does need the help of a little catalyst. So that's just an extra detail, if you like. So right here we have a diagrammatic representation of the hydration of ethene to make ethanol. So essentially we have the OH group attacking the double bond, the H adding across the double bond, and then with the help of dilute sulfuric acid or dilute phosphoric acid as the catalyst at 300 degrees Celsius and about 60 atmospheres, you get ethanol. And this is just another way of writing the same reaction. So the other way you can make ethanol is via the fermentation of sugarcane. So you do a similar reaction modeling what happens on an industrial scale in your labs. However, it's slightly different. So what we'll do is we'll go over this diagram here that kind of overlays what happens on a more industrial scale. And in the next couple of slides, I'll make it a bit more lab focused. So firstly, what we do is we farm the sugarcane, obviously, we harvest it, and then we crush it and dissolve it in water. So basically the sugarcane juice that we've crushed is now dissolved in water. This sugarcane juice is basically rich in two things. So starch and sucrose, and the sucrose is what is significant. So if I grab a pen, the sucrose is what's significant. What you need to know is that sucrose is made up of two things. So it's made up of glucose, which I'm going to draw with the hexagon, and fructose. So it's basically a dimer of glucose and fructose. We then add yeast. So yeast is where the magic kind of all happens. So yeast has two roles in this fermentation process. Firstly, it breaks up the individual sucrose units, so it breaks up the dimer to make individual glucose monomeric units. So you get one glucose and one fructose. So that's the first enzyme present in the yeast. The second enzyme is a bit more interesting. So the second enzyme actually catalyzes the process by which glucose is fermented to form ethanol plus carbon dioxide at a yield of about 15%, which is shown here. So we've got a solution of a lot of mucky things. So we have starchy, a bit of sucrose left over. We've got yeast in it. We've got some glucose. We've got some ethanol, 15% of it, and we've got some carbon dioxide. We've got a little bit of everything. What we can then do to kind of purify it is fractionally distill. So if we distill it, we can get ethanol as pure as 98%. So in this way, we can make sugarcane into ethanol or very pure ethanol by the use of yeast. So this is essentially at a chemistry level what's happening in your labs. So in your labs, you won't start with sugarcane, you'll start with glucose, and via the help of yeast or yeast enzymes, you'll be converting that glucose or C6H12O6. So notice how it's glucose dissolved in water. So we're trying to mirror what happens on an industrial scale. So remember I said that sugarcane was crushed up and dissolved in water, or the juice was dissolved in water. That's similar to what you're going to do. So you're going to dissolve that glucose in water, and with the help of the yeast or yeast enzymes, you're going to make alcohol or ethanol. Make ethanol and CO2, so carbon dioxide. And it's important to also state a couple of reaction conditions. So this has a positive enthalpy. So this is an endothermic reaction. And it's important that you know on an industrial level that although that yeast in a lab level is only catalyzing one reaction, on an industrial level, it's catalyzing two reactions. So it's converting that sucrose into individual glucose units. And then a separate enzyme is converting that glucose into ethanol and carbon dioxide. It's just a small little pedantic detail. So like with all things in chemistry, we need to make sure we maximize yield, especially in this mad topic of chemical synthesis. So I've got a quick summary table showing you exactly how this is going to happen. What I want to do is start here. So we have low concentrations of oxygen. So if you refer back to the previous slide, and I'll clear the slide here, you can see that this reaction has the presence of no oxygen. So we're not using any oxygen or producing any oxygen in this reaction. So this is a totally anaerobic process, and it's really important that we understand this. A couple of reasons why we want it to be anaerobic. If too much oxygen is present, the glucose is converted to carbon dioxide and water instead. So if we have an aerobic process, in the presence of yeast, we won't make ethanol, we'll make water and carbon dioxide, and we don't want that, or like lots of water and lots of carbon dioxide, and we certainly don't want that. Also, any of the ethanol that we do make, if we make any, will be oxidized into ethanoic acid. So we basically will be producing very little ethanol if we let any significant amounts of oxygen into the reaction vessel. The first two dot points are pretty self-explanatory. So we need yeast, obviously, to kind of catalyze the whole reaction. So yeast has a really crucial enzyme that converts the glucose into ethanol and carbon dioxide. And we need a moist environment. So you need a slight little bit of moisture. And that is to ensure the survival of the yeast. Because guys, it's important to realize that this reaction isn't something that happens that quickly. This happens over a couple of days. So usually when you're doing this in the lab, you'll leave it overnight on the Friday, and you'll come back on the Monday and you'll see that you made a bit of ethanol. So the last two dot points are what I feel are the highest yield, because they're what you're most likely to get examined on, especially your practical assessments. So the reason why we like to keep the ethanol concentration less than 15% is because if it gets higher than 15%, it can essentially kill the yeast. So remember that yeast is a biological enzyme. So higher concentrations of ethanol can actually denature or destroy the critical enzymes in the yeast, and thus this process, if I rub it out, so we essentially need yeast for this reaction to occur. However, if we have too high a concentration of ethanol, so if we make more and more ethanol, it can actually destroy the yeast. So this reaction no longer proceeds. So we need to keep the concentrations of ethanol to a minimum. And what we can do is kind of get rid of the ethanol as we make more and more by fractionally distilling it. There are a couple other ways that we can discuss a bit later. Last but not least, it wouldn't be a topic on chemical synthesis if we didn't have a chat about Le Chatelier's principle. So if you look at this reaction, it's got a positive enthalpy. So we're using some of the heat to make the ethanol. So thus, in regards to Le Chatelier's principle, if I increase the heat of the system, that's a change that I've imposed on a system that's otherwise in equilibrium. So the reaction or the system is going to shift in a way to minimize the impact of that change. So thus, if I increase the heat, it's going to use some of that heat and shift from left to right to make more of that ethanol and carbon dioxide. However, there's a slight problem. If I increase the heat too much, or I just blast it with heat, it's actually going to denature or destroy the yeast. So we find a balanced medium. So if I grab my pen, we find a balanced medium of about 37 degrees Celsius for the heat. That ensures that it accelerates the fermentation process, so you get a good reaction rate as well. However, and a good yield as well, because you make more of the ethanol and carbon dioxide, especially the ethanol. However, the heat isn't excessive enough at 37 degrees Celsius, where it can actually potentially destroy the yeast, because the yeast is the kind of tie-in for this whole reaction. So I've got a quick overview of the apparatus, or similar apparatus. So this is a really common setup for an apparatus for the fermentation of glucose to form ethanol. It's important to realize that on an industrial scale, you get the sugarcane, you crush it up, you get the sucrose, and then you add yeast to break that sucrose into glucose. However, on the school level, what you're doing to model that reaction is you're essentially starting with the glucose. So you start with C6H12O6, you mix it in with water, you add some yeast, and then the reaction proceeds. So you don't start at sucrose or the sugarcane. It's important to realize that, especially when you're doing some prac write-ups. However, with regards to this apparatus, we'll go through each of the individual components and state their jobs essentially. So here we have if I grab my pen, we have the water bath. And the job of the water bath is to keep the reaction at exactly 37 degrees Celsius, and it's essentially a water bath, so there's no metal on glass contact, because guys, also recall that this reaction is going to happen over a long time, so days, if not hours at least. So if we have a long-term contact or a long-time contact of glass on fire or glass on hot metal, there's a big risk of that glass shattering. However, with a water bath, what we get is we get a nice predictable temperature at exactly 37 degrees Celsius, and there's very, very, very minimal risk of that glass fracturing and causing damage that way. The next thing we have is our reaction conical flask, which has our reactants in it. So essentially we start with glucose, which we mix in with water, and then we add yeast. So that is the biological catalyst for this reaction. And then what we're going to make is we're going to make ethanol, and we'll leave it just there because the carbon dioxide we do something really interesting with. So as this reaction proceeds, we get glucose, we get water, and essentially the yeast converts the glucose into ethanol and carbon dioxide. As you may recall, carbon dioxide is a gas which is going to bubble away, and it's going to travel up this tube here. And now you might be thinking, what is this secondary beaker here? Why does the tubing empty out from the primary reaction vessel, if you like, into this second beaker with this kind of white substance here? So this is what we call lime water. So lime water, which is calcium CaOH, so calcium hydroxide. When we add carbon dioxide to this lime water, essentially what happens is it forms calcium carbonate, which is white. So essentially, lime water, which is essentially or initially colorless, once that CO2 kind of travels and bubbles into it, forms calcium carbonate, which is white, and thus it gives it a nice milky white texture, right? So in this way, if you initially have lime water which is colorless and you bubble a random gas in and the initially colorless lime water turns milky white, you've proved that the gas that you're bubbling in is carbon dioxide. Last but not least, we have a bung or a stopper here. So the only thing that bung lets out is the plastic tubing. There's a small enough hole for the plastic tubing to fit through, and the reason why we have that bung or stopper, I'll call it a stopper, which is the official name for it, is to keep the reaction anaerobic. And in the previous slide, we went over some of the reasons why we want to keep the reaction anaerobic.